So I wanted to talk about the problems with therapeutic vaccines, what we know about the problems, and what may be other issues that we haven't considered and perhaps we should do. Now, I hope that works. Just to remind you that, that there are real differences between prophylactic vaccination and therapeutic. Prophylactic vaccination is pretty straightforward. What you're trying to do is induce a protective immune response that will prevent infection or control infection when the immunized individual encounters the pathogen. So you give the vaccine before exposure, you generate a protective immune response and memory so that when the individual encounters the pathogen, they get protection and they get no disease, the aim of a prophylactic vaccine. Now you need to contrast that with therapeutic vaccination. In therapeutic vaccination, you are targeting people who already have disease. And they therefore have selected themselves out as a susceptible population because their natural immune response has not been good enough to deal with the pathogen. So the immune response they've made, may not be a good enough one, it may be an inappropriate one, or they may not have made a proper response at all. And your vaccine has to overwhelm that. This is basically turning the tanker around. So it's, by definition, therapeutic vaccines are going to be tough because you've got a selected susceptible population to this specific pathogen. The immune response, if it has been made, is inappropriate or ineffective, and your vaccine has to deal with that. And to remind you, of course, as something I said yesterday, that two arms to the immune response. You've got antibody, which is preventing infection or reinfection, and you've got cell-mediated immunity, which is the immunity you want when you have an established lesion or you have infection. Because the whole purpose of the cell-mediated immune response is to generate interferon gamma and other cytokines that help T cells, such as CD8 positive antigen-specific T cells, to activate macrophages, to activate NK cells, to kill. So it's a killing response, the cell-mediated response, and that's what presumably you want your vaccine to do. As I said again yesterday, the problem is that T cells are much more complicated than Th1, Th2. So you have, in your Th1 subset, they are the warriors of the process, the warriors of the defense process. The T regulatory cells are the diplomats of the process. And regulatory cells are regulating the T cell response. Now, diplomacy is great in most situations, but if you really want a killer response and your regulators are preventing it, then that's a problem. And these are the two issues that are of central importance in therapeutic vaccines. Just to remind you again about the natural course of a genital HPV infection, you do have an immune response. This is a long period, of course. And the immune response of importance, of course, that we want to mimic is the cell-mediated immune response, the successful one. And the cell-mediated immune response to HPV, and these are local responses. So the systemic responses, if you take blood from individuals who either are infected or have been infected, and you try and detect antigen-specific T cell responses, it's difficult because they're at extremely low level, and you really push the assays right to the edge. But if you look at the lesions when they're regressing, such as genital warts, they are packed with immune cells. So there is a profound local response, but the systemic response is weak. However, what we do know from the work of showed van der Berg and his colleagues is that there is memory to E2 and E6 in individuals who've been infected and cleared. And from our own lab, we know that the generation of systemic CD4 antigen-specific 16E2 and E6 responses correlate with the regression of CIN1. And the regression of CIN1 is also correlated with the presence of functional killer cytotoxic lymphocytes in the epithelium. Okay. Now, in the spectrum of cervical intraepithelial neoplasia, I would argue, from the biological point of view, that there are two distinct phases. CIN1 is actually virus replication. It's a complete virus life cycle. All the viral genes are expressed. CIN3 is a totally different beast. The virus life cycle is incomplete. Only a subset of viral genes are expressed, and there is deregulation of viral gene expression. Now, the therapeutic efforts have been very much targeted to high-grade disease, to CIN3, rather than targeting it to infection. And this targeting it to high-grade disease is another difficulty. Because in cancers and most high-grade lesions, the E6 and E7 genes, of course, are constitutively expressed. So, and the continued expression of these oncogenes is essential to maintain the transformed phenotype, as it were, in vitro, and maintain the malignant phenotype in vivo. So if you're thinking about a therapeutic vaccine, the only two possible targets are E6 and E7, because these are the only viral proteins that are going to be expressed in all high-grade intraepithelial neoplasms caused by HPV and their associated cancers. Now, this deliberate immunization with 16E6 and or E7, another oncogenic E6 and E7s, and the generation of antigen-specific cytotoxic T lymphocytes has been tested, and it's been tested in a very large range of potential vaccine delivery systems. And most of these studies use a transplantable HPV-positive mouse tumor model or models. Well, I have very good news for mice. And the very good news for mice is that HPV-expressing cancers are relatively easy to cure. I unfortunately have very bad news for humans, in that the HPV-induced cancers have been largely refractory, and indeed the high-grade precancers have been largely refractory to the approaches that have been successful in mice using these models. Now, why is that? Well, you have to ask what your HPV mouse tumor model is. In most of these situations, you take mouse cells which are expressing E6 or E7, you inject them into the mouse in large, say 10 to 6, 10 to 7 cells. Within 2 weeks, there is a tumor forms at the site of injection. You then use your vaccine to see whether that tumor will regress. Now, this is completely different to the biology of tumors developing in humans, because the development of a tumor in humans and in natural development in animals is a Darwinian phenomenon. So the cells become abnormal. Most of these abnormal cells would be actually identified by the immune system, both innate and adaptive, and cleared. But the genetic instability that is a feature of these tumors results in the ability to evade the immune system, a phenomenon known as immuno editing. So there is escape from the defense systems, and the tumor becomes independent. Now, this does not take place overnight. It takes place over a long period of time. So you've had a long period of acquisition of genetic changes which allow the tumor cell to modify its environment and to evade tumor-specific killing. So you end up with something like this, and I know Sjord will be confronting many of these issues, because your tumor cell downregulates surface molecules which could be recognized by the immune system. It generates cytokines and soluble factors that modulate the systemic immune responses and modulates other cells coming in. The microenvironment, because remember the tumor cell alters the vascularity, it alters the connective tissue cells to provide a stroma which is suitable for the tumor itself. These are functionally impaired. So you have inhibition of cells that could kill, you have recruitment of cells which are regulating the killers, such as T regulatory cells, you have inhibitory receptors on the tumor cell. So here you've got a huge problem to overcome. And if you look at disease progression in cervical neoplasia, you're seeing exactly that. In CIN1, you've got a delay in activating HPV-specific cell-mediated immunity, but it is still possible because you've got a regular viral life cycle. As soon as you go into the high-grade disease, you have an immunosuppressive milieu, you have immature dendritic cells, antigen-presenting cells, you have cytokines that induce tolerance, such as TGF-beta. You downregulate the cytokines that would be effective, the chemokines and the receptors on the vascular channels which attract immune cells, and you have a dominance of T regulatory cells. And all this is an acquisition as you go through the grades of cancer. And so if I go back to the cell-mediated immunity to HPV, progression in the CIN spectrum is correlated with the absence of intraepithelial CD8-positive T cells. This actually was shown by us rather a long time ago when we used an antibody in the early 90s, which is actually against the alpha-4 beta-7 integrin, and showed that there was a removal of intraepithelial T cells expressing this surface marker in CIN3. And Connie Trimble, in what I think was a seminal paper in the Journal of Immunology in 2010, showed that there certainly was a depletion of these cells in CIN3. And she showed that the stromal endothelial cells, the cells on the endothelial cells lining the vasculature underneath the lesion, lost the ligand, MADCAM1, the ligand for the alpha-4 beta-7. And that this loss correlated, was associated with CIN progression. This was a retrospective study. So that rather argues that even though you generate these cells, the microenvironment of the tumor is actually preventing their recruitment into the abnormal epithelium. And so I think what we know, or what we recognize now, is that if we are going to generate a successful therapeutic vaccine, first of all, it's got to be highly immunogenic, and that is the value of the mouse models. The mouse models give you proof of principle that your vaccine candidate is capable of generating a strong T-cell response. And so we mustn't downgrade those, because that's an important phenomenon. The immunogenicity may be enhanced by the appropriate adjuvants. The many strategies actually target antigen to the antigen-presenting cell, to the endosome. You have DNA vaccines for HPV and for other papillomaviruses, which if you codon modify, you increase protein production. So all that, I think, is entirely doable. Those are things that we know what to do. But as I've said to you, this is not just about the tumor cell itself. It's about the tumor and its environment. And the local immune microenvironment has to be manipulated. This could be achieved by immunomodulators and adjuvants that target innate immune receptors. It could be possibly by identifying those receptors on the connective tissue or the vascular channels that need to be upregulated or downregulated. And you also are going to have to regulate T T regulatory cells, because this seems to me to be one of the key challenges of therapeutic vaccines, because some work published from the Van der Berg laboratory showed that their quite successful therapeutic vaccine can't clear large lesions, and it can't clear them because of T regulatory dominance. And the other thing I think that's come out of the various trials that have been done is that the therapeutic responses are not immediate. They accumulate over time. So you are seeing regression of lesions after 12 months. You're not seeing them immediately. And we may well need to rethink our interventional strategies because of that. And I want to say that there's another set of complete unknowns which we simply haven't addressed. Now, HPV-associated cancers tend to arise at squamocolumnar junctions. These are sites, of course, of epithelial instability because you've got squamous and glandular epithelium meeting at a junctional point. Now, what we don't know is whether the biology of these junctions is the same at all the anatomical sites. We do know that the cofactors are slightly different for each of these anatomical sites for the cancers. So the cervix, if you look at transgenic mice, which have been used as a good model, in the cervix you need estrogen blockade to get the progression from expression of E7 through the various stages of the precancers to full cancer. In the anus, in addition to expression of the oncogenes, you require carcinogens. In the oral cavity, you also require carcinogens. These are in the transgenic mice models. The epidemiological studies tell you that in the cervix, co-infections are important. Oral contraceptives are a risk factor. Smoking is a risk factor. In the anus, immunosuppression is a risk factor. And in the head and neck, chronic inflammation appears to be a risk factor. So I think that this has implications for understanding, first of all, the natural immunity in these sites, but also how you might expect your therapeutic vaccine to work. Because we know the generic mechanisms for HPV-mediated oncogenesis are the deregulation of HPV-6 and E7 expression. We know cells at squamocolumnar junctions are targets, but the biology at these junctions, what the cofactors are, what the microbiome, the commensals at each of these sites may be playing an important role in regulating immune responses. We know very little about that. We don't know whether the target cell is the same at each of these sites. And these are all research questions, but it does put up a big unknown. Will the same therapeutic approach be effective at the different sites? So I think what I've said to you is therapeutic vaccines are tough. But I have to say that I think our next speaker has really challenged this tough concept. Thank you very much.